Hi, it's Miss Parrott, and this is video number 6 in the series AP Biology Equations Explained. Today we're going to be tackling Hardy-Weinberg by tracking evolution with allele frequencies. Be sure that you're up to speed with your genetics vocabulary. Check out my genetics fundamentals video in the description box below. So AP Biology defines evolution as a change in allele frequency over time. And what Hardy-Weinberg allows us to do is answer a question like, if this was the allele frequency in the past and this is the allele frequency now, is evolution happening? If the allele frequencies are the same, the population is not evolving. But if the allele frequency is different, then it is. Now let's break down the formulas. The formulas can be found right here on your AP Bio equation sheet, along with the definitions of P and Q. So before we break down the equations, know that in Hardy-Weinberg equations, we assume that each gene has just two alleles, a dominant one or a recessive one, or they may be incomplete or codominant, but the point is there's only two alleles. We know that reality is a lot more complicated than that, but we're doing a basic model. Also, when we're doing Hardy-Weinberg problems, the frequencies are written as decimals. So even though it's easier for our brains to think about percents. So in 100% of a population, 50% look like this, and the other 50% look like this. We would say 0.5 have this and 0.5 have that. So instead of percents, we're just going to convert that to a decimal for what we're doing today. And that way, when you combine them all together, 100% as a decimal equals 1. So with that understanding, let's look at the first Hardy-Weinberg equation, which is P plus Q equals 1. Meaning P is the frequency of allele 1, most often the dominant allele, plus Q, the frequency of allele 2, most often the recessive allele. You combine those frequencies, that's all the organisms, that's 100%, so that equals 1. Similarly, if you look at the second equation, we're going to be using P and Q to look at frequencies of different genotypes. So there's 3 possible genotypic combinations. Homozygous dominant, so that's big A, so that would be P times P, which is P squared. So we represent the frequency of the homozygous dominant genotype as P squared. The frequency of the heterozygote, big A, little a, is 2PQ. Why not just PQ? That's because if we look at a Punnett square, let's say we mated 2 heterozygous individuals, the chances of having the heterozygous genotype are twice that of either of the others. So we do 2, because 2 boxes have big A, little a, 2 times P times Q. That's the frequency of the heterozygote. And then the frequency of the homozygous recessive, or little a, is Q squared, right? Q times Q is Q squared. And that's going to equal 1. Why? Because think of 1 as 100%. That's all the individuals with all their genotypes together in the whole population. So Hardy-Weinberg gives us, or the Hardy-Weinberg equilibrium gives us a baseline for what does the population look like if no evolution is happening? What are the current allele and genotypic frequencies? So we have 5 assumptions that must be true for Hardy-Weinberg equilibrium to be established. 1, we need a large population. 2, no migration. 3, no mutation. 4, random mating, meaning no individual's more attractive or has more reproductive success than the other. And 5, no selection. So no trait gives a greater chance at being successful in survival and reproduction. So these, I don't know a population in the whole wide world that meets those criteria. So, I mean, yes, Hardy-Weinberg equilibrium in a population is basically impossible. But what's the point? It gives us a baseline from where to start to see if over time our population is changing. Let's work a practice problem. So, in this example, in cave salamanders, 16% show the recessive phenotype. If there are 200 salamanders, how many individuals are there of each genotype? Okay, so we have information in percent form about the recessive phenotype, but it's asking us how many there are of each genotype. So how do we go from phenotype to genotype? Well, luckily, it's giving us the 16% show the recessive phenotype. Remember, that's our little a is going to be the genotype that pairs with the recessive phenotype. So there's only one genotype that could possibly have that phenotype, and that's little a, which, hey, is the genotype that corresponds with Q squared. So let's not forget, instead of percentages, we write percents as decimals. So based on the question, we know that Q squared equals 0.16. That's 16 as a decimal. Now we're going to determine Q, or the frequency of the recessive allele, by taking the square root of Q squared. So the square root of 0.16, if you put that in your calculator, don't forget you need to be using the calculator that you plan to use on the AP exam so you know where your square root button is. That's going to be 0.4. So 0.4 equals Q. So Q squared, 0.16, Q is 0.4. Now we can use our first Hardy-Weinberg equation to find P. So the first equation is P plus Q equals 1. So we'll write that P plus, we just found Q is 0.4 equals 1. We're going to isolate P by subtracting 0.4 from each side. So P equals 0.6. All right, we have our allele frequencies. Step number 4 is to find the genotypic frequencies. So we're going to use our second Hardy-Weinberg equation, P squared plus 2PQ plus Q squared equals 1, and the allele frequencies from just before. So if P, we just said, was 0.6, 0.6 squared equals 0.36. That's the frequency of the homozygous dominant genotype. 2PQ, we'll do 2 times P, which is 0.6, times 0.4, which was Q, and that's going to give us 0.6 times 0.4 is 0.24, times 2 is 0.48. Of course, you can do this in your calculator, but sometimes it's easy enough to do in your head. And then Q squared, right, was given at the beginning of the problem. If you weren't sure, though, you could do 0.4 squared, or you could take 1 and subtract 0.36 and 0.48, and you would get 0.16. So a little algebra, and you're good. Finally, you're going to reread the question and answer what is it asking, because more often than not, it doesn't want you to stop right here. So the question asks, if there are 200 salamanders, how many of them are there of each genotype? So here are our possible genotypes. We're going to take our frequencies and multiply by the total number of individuals to figure out like what percent of the individuals have each genotype. So our P squared was 0.36. our heterozygous 2pq was 0.48. Our q squared was 0.16. And if we do that math on our calculator real quick, 0.36 times 200 is 72 individuals. 0.48 times 200, that's probably going to be 96. Yep. And 0.16 times 200 is 32. So 72 individual salamanders we would expect to be homozygous dominant. 96 individuals would be heterozygous, and 32 individuals would have the homozygous recessive genotype and therefore phenotype. So what next? A question like an FRQ or even a multiple choice question, it may give you you have to figure out the current data, and then it might give you a later generation's data, and it'll ask you, is the population evolving, right? And if P and Q remain consistent or relatively consistent, then no, evolution is not happening. If P and Q are different, then yes, the population is evolving. It might also follow up with a chi-squared test to compare observed versus expected values. I'll actually be doing an example of a video like that a little bit later this week. Or an FRQ may follow up and ask which of the Hardy-Weinberg assumptions is being violated that leads to this evolution in the population. And you have to come up with some reasonable answer that would involve that maybe instead of a large, you have a small population, or migration either in or out of the area is occurring, that a mutation occurred, or that mating isn't random, or perhaps that one of the phenotypes has a selective advantage over the other. Common errors students make that you want to be sure you don't make are mixing up P and P squared or Q and Q squared, forgetting to turn your percent to a decimal. So if when you add everything up at the end of your P's and Q's, or P squared, 2PQ, and Q squared, if it equals more than 1, and I'm not talking 1.01, I'm talking like 14, then you know you've done something wrong and you need to go back. Also, you shouldn't round before the final answer. That can give you some iffy answers. And then finally, another common error is to assume the recessive phenotype gives you Q instead of Q squared. Remember, a recessive phenotype gives you the corresponding recessive genotypic frequency, that's Q squared. So to wrap things up, key takeaways, 2 equations, lots of big insights. Hardy-Weinberg lets us predict genotypic frequencies and test whether or not a population is evolving. If this video helped you, don't forget to like, subscribe, and comment or ask a question about Hardy-Weinberg in the box below. Check out my other playlists, the rest of this whole AP Bioequations Explained, my genetics and evolution playlists. And teachers, feel free to check out my resources on Teachers Pay Teachers at Know and Grow with Gnomes. Keep learning, keep growing, and we'll see you next time.